Hi everyone. In tonight's video, I'm going to talk about three different types of lipids: triglycerides, phospholipids, and cholesterol. They each have a different structure and function to the cell. So the first one I'm going to talk about are the triglycerides. These are commonly called fats. They're composed of one glycerol molecule. So you can see here is our glycerol with the 3 carbons. And again, with the 3 carbons in this model of glycerol, and then 3 fatty acids, which are highlighted in green. So I have 1, 2, 3 fatty acids in each of my triglycerides pictured here. A triglyceride is formed by 3 separate dehydration synthesis reactions. So to form each triglyceride, you take 1 glycerol and 3 fatty acids. Each one undergoes a dehydration synthesis reaction, and that produces 3 water molecules to get your fully complete triglyceride. The function of a triglyceride is energy storage. Again, there's a difference between energy storage and an energy source. Triglycerides have to actually be broken down into individual 2-carbon units called acyl groups in order to be an energy source. So these are simply energy storage. Triglycerides are stored in cells called adipose cells or fat cells, and these are pictured here. So this is what an adipose cell looks like if you look at it under the microscope. You see a nucleus shoved to the side of the cell, and the whole interior of the cell is composed of this fat droplet, which is full of triglycerides. So remember from a previous video that fatty acids can be either saturated or unsaturated. I want to talk about saturated fatty acids and how they impact the structure and function of triglycerides. These are made by animals. So the triglycerides made by animals have saturated fatty acids. That means they have single bonds between all the carbons in the fatty acid. These single-bonded saturated fatty acids have a very straight shape, like is pictured here, and that means that they pack very tightly together. So they're a solid at room temperature. So a good example of a fat or a triglyceride made by animals is butter, which is pictured here, which is of course solid at room temperature. What about triglycerides that have unsaturated fatty acids? And remember, in nature, when natural systems make unsaturated fatty acids, they generally make them in the cis conformation. That gives them a very bent or kinked shape. These are made by plants and fish. They have 1 or more double or triple bonds between the carbons in those fatty acids. And again, that gives them this bent or kinked shape. That means they kind of loosely pack, because there's all this space in between the fatty acids due to the kinking or bending of those fatty acid tails in the triglyceride. This makes them a liquid at room temperature. They are oils because they are more free to move around because they have this extra space. A good example of an unsaturated fatty acid is olive oil, which is of course made by olives, which are plants. We have an abbreviated structure for each of these forms of a triglyceride. So if I wanted to draw an abbreviated structure of a triglyceride that had saturated fatty acids, I would draw this rectangle at the top to represent the glycerol part of the triglyceride, and then 3 straight lines to indicate the saturated fatty acids. If I want to draw an abbreviated structure for an unsaturated fatty acids, I would again draw the rectangle for the glycerol, but I would intentionally draw the lines indicating the fatty acids with bends or kinks. So what we have found is unsaturated fats are a far healthier part of our diet. They don't clog our arteries. So scientists had a great idea. They thought, what if we convert these cis unsaturated, which are very healthy fats, to saturated form? The idea for wanting to do this is that they would be healthy and they would be a solid at room temperature, and it turns out they have a much longer shelf life. So what scientists did is they took cis unsaturated fatty acids, which are very healthy, and they put them under high temperature and high pressure, and that's called chemical hydrogenation. What they were trying to get was these saturated fatty acids, but hoping that they would still be healthy because they were made by our plants. This was the goal. What they actually got were these incomplete hydrogenation. So what they got was adding some hydrogens, but when they did, they came in the trans configuration. They became what are we called trans fats. And trans fats, although they were solid at room temperature, think margarine, and they did have a much longer shelf life, so they accomplished that. But it turns out they're very, very unhealthy. So if we look at this diagram here, our unsaturated fats that are made by plants and fish are very healthy. So avocados are a very healthy source of fat. Saturated fats, which are made by animals, an example is bacon, are very, very unhealthy. And trans fats, which are an artificially induced partial hydrogenation, they did accomplish the goal of being increasing the shelf life, but they've turned out to be very unhealthy. As soon as we started using a lot of trans fats in our diet, we found that there was an increase in cholesterol problems and diabetes. And so now any food that has trans fats in it has to have it clearly labeled on its label because they have found to be so unhealthy. The next lipid I want to talk about are phospholipids. Phospholipids have a very different structure. They are again composed of a glycerol unit. Right here you can see our 3 carbons. And then on top of that glycerol unit is attached a phosphate group, which you should remember from when we covered our functional groups. So this glycerol plus the phosphate is what's referred to as the head of the phospholipid. So here it is in a diagram, and here it is in more of a chemical form. This is the head group. It's the glycerol and the phosphate. Instead of having 3 fatty acid tails like the triglyceride, the phospholipid has 2 fatty acid tails. Each is formed by a dehydration synthesis reaction connecting to this glycerol. The third carbon that would have the third fatty acid in a triglyceride is bonded to the phosphate, so there's no room for a third fatty acid here. So we call this head group the phosphate and the glycerol, and we call the fatty acids fatty acid tails. These fatty acids can again be saturated or straight, or they can be unsaturated or encanked. We also have an abbreviated structure for these phospholipids, just like we did triglycerides. And so we draw a round head unit, which is the glycerol and the phosphate, and then we draw the 2 tails. When you are drawing this abbreviated structure, you should draw the tails very carefully. If you are indicating a saturated fatty acid, you want to draw a straight line. And if you're indicating an unsaturated fatty acid, you want to draw a very curved or kinked line. This particular model is somewhat ambiguous because you can't really tell if that's straight or curved. I would advise you to draw them very clearly with a lot of intention. The function of phospholipids is structural. They're not an energy source or energy storage. They are components of our cell membranes. So looking at a phospholipid molecule, is it polar or nonpolar? Well, it turns out it's both, because this head group that has the phosphate and the glycerol is very polar. It's hydrophilic. It dissolves in water. The fatty acids are very hydrophobic. They are nonpolar. They do not dissolve in water. This leads to phospholipids behaving in a very unique manner if they're placed in water. If you put phospholipids in an area where there's both oil and water, the head groups will arrange themselves so that they are in the water because they're polar, they're hydrophilic, and they'll dissolve in the water. The fatty acid tails will arrange themselves so that they're in the oil because they're hydrophobic, they are nonpolar. What about if we put phospholipids in all water and there was no oil? They often arrange themselves in these what we call bilayers. So the hydrophilic head groups are all facing the water because that's where they dissolve. And the tails arrange together, forcing the water away so that in the middle here between this layer of phospholipids and this layer of phospholipids, you have a very hydrophobic region. I can show this up close a little bit more, where this is how phospholipids will arrange themselves in a bilayer when placed in water. So we have our polar head groups facing water on either end, And in the middle, we have these hydrophobic tails excluding water. So cell membranes are composed of these phospholipid bilayers. So we can look at this cutaway of a cell. Here is the interior of the cell, which tends to be very aqueous and full of water. So the hydrophilic head groups of the phospholipids are facing the interior, and they're also facing the exterior of the cell, which is in an aqueous environment. In between the bilayer, which is where the fatty acid tails are, it's very hydrophobic. But our cell membranes are composed of these phospholipid bilayers. The last lipid I want to talk about tonight is cholesterol. Cholesterol is composed of this 4-ring structure. It doesn't look anything like a phospholipid or a triglyceride. So why do we classify it as a lipid? Well, if you remember from a previous video, lipids are classified as anything that doesn't dissolve in water. That's a pretty broad definition. So some examples of these cholesterols are cholesterol itself, cortisol, and testosterone. These are all steroids. I'm going to focus on cholesterol itself. Cholesterol's function is it can be a precursor to some of these hormones, but it mainly functions in membrane fluidity in animal cells. And when I say mainly functions, it doesn't actually mainly function in the cell. This is the main function I am going to be talking about in tonight's video. So what we see is that cholesterol can kind of intercalate in between the fatty acid tails in a phospholipid bilayer, and its function is it helps keep animal membranes fluid at low temperatures and maintains their structure at high temperatures. This all has to do with the fact that animal membranes tend to be composed of phospholipids with saturated fatty acids, so they're linear, so they pack tightly together, and you need cholesterol to kind of give them some elbow room to maintain their integrity and their fluidity. This is covered in much more detail in another video on my YouTube channel called Introduction to Membranes. So that's all for tonight.